In this unit, you've seen lots of pieces of information that come together to provide a case for the pattern of evolution and descent with modification. And in your prep assignment, I've asked you to organize this evidence in terms of these contrasting predictions that arise from descent with modification as compared to creation. And the purpose of this web lecture is to give you a model for how to do this, how to fit these pieces of evidence into these different categories. But before we start on that, I want to talk a little bit about what do we mean when we say evidence? What do we mean when we say proof? What are we really talking about here? Merriam-Webster Dictionary defines evidence as something which shows that something else exists or is true. A visible sign of something. So the visible manifestation of something that is invisible or is not seen for whatever reason. And so this evidence, these signs, can be weak or strong, or they can be more or less convincing. One way in which we typically think of evidence is in a legal context, right? When we're solving crimes. And so if you are, for example, in a murder trial, and your only evidence is that a neighbor heard a fight, heard a voice calling out, "I'm going to kill you," it seemed to be the defendant's voice, and then a shot, that's kind of weak evidence to base this murder case on, right? Or maybe you could have a neighbor who saw the person through the window, looked like the defendant, and saw them pull the trigger. That would be much stronger evidence. So we've got this range of evidence. Both of these are signs, right, of something that you actually can't see because it happened in the past. But one of them is really more convincing, having actually seen the person versus just having heard a voice. Both evidence, but not as strong. And so we can have the most confidence in multiple pieces of evidence that come from different sources that are not directly related to each other. So in our murder case, for example, maybe one witness, a neighbor, heard the voice, another one saw the person through the window, the police found the defendant's fingerprints on the gun, and those are all pointing toward the same answer, that the defendant is guilty. But does that mean that this is the only possible thing that could have happened? Does this mean we're 100% sure? No, that means that we have pretty good confidence that this is what happened. If we had more evidence, we'd be even more confident. But can we be 100% sure? No. Is it at least possible that some other guy who kind of looked like the defendant and sounded like the defendant stole his gun so it had his fingerprints on it and then committed the crime? Sure, it's possible. Do we think that's the best explanation for the evidence that we have? Probably not. So we can't be 100% sure, but maybe we have enough evidence to be convincing. And this, in common language, is what we think of as proof, having enough evidence that we're convinced that something is true. But as rigorous thinkers, we don't really like definitions that may not be applicable to every situation. So a body of evidence that is very convincing to me might not be very convincing to you. You really think that there's another explanation that seems plausible. So if we're going to have any kind of objective definition of proof, I think it would have to be that you've proven something if there is no other possible explanation for the observations. So this comes down to a logic problem, a logical argument. The catchphrase I use is that proof is for mathematics. This situation where there's no other possible explanation, that the possibility of that happening in the physical world is vanishingly small. So as biologists, we tend to speak in terms of how much confidence we have in something rather than saying that we've proven something. So banish the word proof from your vocabulary for this class. We want to talk about how confident we are in some sort of theory evidence. What I want you to understand from this whole unit where we're looking at the evidence for evolution is that there's a lot, and it comes from lots and lots of sources that are not directly related to each other. And so because of all of this evidence, biologists are extremely confident in the theory of evolution, as confident as any scientist is in any theory, germ theory, atomic theory, plate tectonic theory, gravitational theory. We can have very, very, very high confidence that these theories correctly describe the way the world works. And usually when science changes its mind about something that it has high confidence on, it's usually because it turns out that that thing is more complicated than we originally realized and we need to add to it. Not that we need to throw it out because it was wrong, but it's incomplete in some way. So we can be very confident that species do evolve over time. They are transmutable in that old-fashioned early evolutionary thinking language, and that natural selection is the mechanism by which species adapt. So the way we do this in science is we take two competing models to explain a phenomenon, such as the diversity of life on Earth, and we use those models to generate predictions about what we expect to find in terms of evidence, and we see where the evidence falls. So the two competing models, as we've discussed in class, in this case are special creation and descent with modification. And we've discussed also in class these specific and completely contrasting predictions that each of these models makes about what we expect to find. So one is that special creation predicts that species don't change over time, whereas descent with modification predicts that they do. Special creation predicts that lineages do not split, whereas descent with modification predicts that they should split and diverge. This is what we know as speciation. Special creation predicts that each species is separately created, whereas descent with modification predicts that new life forms are derived from older forms, so macroevolution. And special creation predicts that each species is independently created, whereas descent with modification predicts that all life forms are related to one another through common ancestry. And special creation predicts that Earth and life are young, whereas descent with modification absolutely requires a large span of time for these minute changes to accumulate over time, and so Earth and life would have to be very, very ancient. So let's look at each one of these contrasting predictions in turn. So let's begin with the first prediction from special creation, that species do not change over time. So remember, we defined evolution initially as changes in heritable traits over time. So, and it's one of the very common arguments you hear from people arguing against evolution is that if this is such a slow, gradual process, how can we ever know that it's real? Can we ever actually observe this in the world? One of the pieces of evidence, one that you're very familiar with at this point, is the case of the soapberry bugs. If you have not already watched the video on soapberry bugs, go ahead and stop this one and go back and switch to that one and get caught up. But I'll just quickly go over the short version of the story. So, as you know, the South Florida populations originally fed on these fat balloon vine fruits that you see here at the bottom, and had very long beaks for poking through these seed pods and getting into the seeds within. Recently, a close relative of those balloon vine plants started to be planted in central Florida as an ornamental, and these soapberry bugs from southern Florida started to begin to come into these areas where there was the golden rain tree fruit with the smaller seed pods. And now what we see in central Florida is the bugs in central Florida began feeding on those plants, and now they have short beaks. So we've got a story that these populations of soapberry bugs in the southern part of Florida that feed on the fat fruits have long beaks. The ones in central Florida that eat on the flat fruits have short beaks. But have we really identified a change over time in a heritable trait? As you know from the video on soapberry bugs, scientists have done reciprocal transplant experiments where they've taken bugs from both populations, raised their eggs on both kinds of fruit to see whether the length of their beak depended on which population they were from, or if it depended on which fruit they fed on. And it turned out that they depended solely on which population they came from. Soapberry bugs that were from the Key Largo population had longer beaks regardless of whether they were fed balloon vine or golden rain tree pods, and ones from the Lake Wales population had shorter beaks regardless of which fruit they were fed on. So this is clearly a heritable trait. And so the graph you're seeing here is measurements from museum specimens from collections at different points in history in Florida, with beak length plotted on the y-axis and the date at which the bugs were collected on the x-axis. And you can see that prior to the introduction of ornamental golden rain trees in central Florida, there was the full range of beak lengths. Soapberry bugs, they had very long beaks. And then around this time, around 1950, somewhere around here, In central Florida, we start to lose this upper part of the distribution, and we see more and more with the short beaks. Why? Because remember that the range of these balloon vine plants was shrinking and shrinking and shrinking as this other plant, ornamental plant, became invasive and started encroaching on its range. So you get fewer of the long-beaked bugs that feed on the balloon vine plants. And again, we can use Covent Garden experiments to confirm that this is a heritable trait and not due to the effects of feeding on different foods. So over a relatively short period of time, we can see over the past 50 or 60 years, this change in a heritable trait. And granted, this is just a few millimeters on a beak, but it's, again, only 50 or 60 years. So when you have strong selection pressure and an organism with a relatively short generation time, you actually can observe these changes over time. And if 50 years can make a measurable difference in beak length, think what billions of years could do. The second prediction made by the special creation model is that lineages do not split, whereas the descent with modification model, we predict that the diversity that we see in life is produced by speciation events. The diagram you see here shows our understanding of how the speciation occurs. So beginning with a single population with a large amount of variability. So you see in these fishes there are whitefish, greenfish, and bluefish all living together in a single population. At some point, something happens to split this single population into two populations. And these two populations are going to diverge in some way. So, for example, these fish might be living in the region of Panama before the Isthmus of Panama formed, and then the land bridge formed geologically, and these fish became separated. And maybe on this side of Panama, it's mainly sandy beaches, so these white fishes do better. Let's say perhaps on the other side, for example, there are mostly seagrass beds, and these green fish do better at hiding from predators. There's still maybe some of these bluefish around in the population, but you're starting to see a divergence where natural selection is favoring one of these variants on one side, another one on another side. And over time, these fish diverge both morphologically and genetically to the point where eventually there's a complete split. And if these two populations were to come back into contact, they would be reproductively isolated. And so that's where, at the point at which we would call this a true species. So, in terms of finding evidence for this process, it's very tricky, right? This is a very lengthy process. Even with a fairly short generation time, it takes many, many, many generations for this to happen. And also, this point at which they become two separate species is very hard to define in the real world. It's a continuum, and there's no single point at which you could say, aha, now we have two different species. But we can make the prediction that if this model is correct for how speciation happens, we should be able to find populations or pairs of populations that represent any of these particular stages of speciation. And it turns out that we can do that with groups of kind of fish called the three-spined stickleback. The first population we're going to consider lives in a place called Roberts Lake in Canada. And this population has two different feeding strategies. It can feed either up in the water column, and these forms have these structures called gill rakers surrounding their throat that helps to filter food out of the water column. And so these fish that tend to feed more in this way have long gill rakers. Ones that tend to feed more on things like insects and large pieces of food tend to have shorter gill rakers. And we see in the population as a whole, we've got a normal distribution of gill raker length. And so that would represent this first stage where you have a single variable population. If you look in that same lake and also in the creek that's the outlet for the lake, you can see that there's a very distinct creek form that's larger in size and has short rakers. It tends to feed on these bottom-dwelling prey items, whereas the ones that are in the lake tend to be more this water column feeding form with long gill rakers. But if you look at the genetic differentiation between these two groups, it's really a continuum as you go from the lake to the creek. They interbreed readily, and there's a whole hybrid zone between the two that shows that these two are still readily interbreeding. In a different lake, you begin to see even more differentiation between species of three-spined stickleback. So again, we've got a lake bottom feeding fish that is larger in size and has short gill rakers, and then another form in the population that lives in open water that's very small and has long gill rakers. These two variants in the population, they can interbreed and produce viable hybrid offspring, but those hybrid offspring are not particularly successful in either part of the habitat. They're not very well adapted to either feeding on the bottom or feeding in the water column, and they tend to be outcompeted by the normal forms for that area. So there's a form of reproductive isolation in which the intermediate forms just don't survive very well, and that would represent this stage in the speciation process. Yet another group of three-spined sticklebacks living in the Japan Sea. They're very morphologically distinct. You could readily identify them as different fish. They do overlap in habitat, and they do sometimes interbreed with each other, but the offspring that they produce are sterile. So we do see evidence of different populations that seem to be in different places along this continuum. The third prediction that arises from the model of special creation is that each species is separately created in its own individual act of creation, whereas the descent with modification model says that species arise from older species, that everything is related in a great tree of life, each species coming from one that came before it. And our best evidence for this is really transitional species in the fossil record. So what is a transitional species? So a transitional species is an organism that has features that are intermediate between its ancestor and its progeny. So it's going to have some features of older groups that it came from, and also some features of living species that it gave rise to. So the transitional species that you're already familiar with from your reading is Archaeopteryx. This was a fossil that was found just a couple years after the publication of The Origin of Species. Thomas Henry Huxley was amongst the first to note the similarities in features between modern birds and the dinosaur fossils that they had at the time. But this was the first real transitional form that really had features of both. So it had feathers and wings like a bird, but it also had a long tail, which no living bird has, but it's very common in reptiles. And it also had teeth, which no living bird has, and obviously reptiles do. And we now know that this is one of the early branches of the clade that led to birds. And since then, we've found many, many, many other fossils of feathered dinosaurs, and we see that Archaeopteryx is just one in this whole family of transitional forms between the dinosaur ancestors and the bird descendants. And a really interesting thing about this idea of transitional fossils is that it gives us some predictive power of what we should expect to see in the fossil record. We can make predictions about intermediates that might have existed between two forms and actually go out and look for the fossil evidence of those transitional forms. And an interesting example of that is in turtles. So turtles are extraordinary among vertebrates for many reasons. I'm sure you're aware that they're very unusual. But one of the really unique things about turtles among vertebrates is that every other tetrapod vertebrate, vertebrates with 4 legs, have ribs that, as they develop, they follow the contour of the body wall. And so because of this, they stay inside the shoulder blade as they develop. So we have ribs on the inside, and then the shoulder blades come around to the outside of them. Turtles, on the other hand, have had a developmental innovation so that as their ribs develop, they don't follow the contour of the body wall. They spread out externally and they sort of push the body wall out of the way. And as they develop, these anterior ones toward the head kind of push over and go over the top of the shoulder blade, which you see here. And so this is an earlier stage of development where you can see the ribs coming to the outside of the shoulder blade instead of staying to the inside of the shoulder blade. And then in later development, they sort of spread out and fuse with the outer shell. And so we've got two distinct patterns there, one where the ribs stay inside the shoulder blade and one where they go outside. We should be able to find some sort of early transitional turtle that has some intermediate in those two features. And in fact, such a transitional fossil has been found. This is a fossil of an ancient turtle. What they found doing an analysis of the bone structure of this fossil is that the ribs don't come and fan over. They still ignore the contour of the body wall. As you can see, these ribs are straight out and flat as compared to a typical tetrapod or amniote vertebrate where they curve along the body wall. But they don't fan forward like in modern turtles to cover and go over the top of this shoulder blade. They're sort of lined up alongside of it. So this is another example of a transitional fossil that's intermediate between these amniote ancestors, this feature that's shared by all amniotes, and so we know it would have been present in the ancestor of turtles and the living descendants, which are the turtles. The fourth prediction made by special creation is that each species was independently created in a separate act of creation, and so you'd expect each organism to have its own blueprint well suited to what the creator had in mind for that organism's role to be. In contrast, descent with modification tells us that all organisms are related to each other in this great tree of life, and there are going to be similarities between organisms based on common descent from a common ancestor. And the best evidence we have for that comes from comparative anatomy and looking at what we call homologous structures. So the picture that you're seeing here are the forelimbs of several mammals, and what you see is this pattern that you're now familiar with from watching Your Inner Fish of one bone, two bones, a group of small wrist bones, long hand bones, long finger bones. And you see this in every single tetrapod you'll ever meet to the extent that they have limbs. The same pattern no matter what they're adapted to do. So a mole forelimb that's adapted primarily for digging. Again, one bone, two bones, wrist bones, hand bones, finger bones in that order. Horse limb adapted for fast running. Again, the single humerus. In this case, one of the two bones is highly reduced and becomes part of this much stronger bone, but you can still find the elements of the two bones there. Wrist bones, hand bones, finger bones. Dolphin, adapted for swimming, again, one bone, two bones, wrist bones, hand bones, finger bones. A bat, whose forelimb is adapted for flying, same pattern, one bone, you can see the second bone just teeny weeny here. One bone is better for strength, and so the second bone becomes reduced or incorporated into that one bone, but you can still see the homologous structures. Wrist bones, again, fused for strength, but definitely homologous. Hand bones, finger bones. And this pattern would be really, really unexpected if each of these organisms were separately, independently created for whatever job they're meant to do. So imagine that you are an engineer and you've designed a really good boat, let's say, and you then want to go and design a really good airplane. And you think to yourself, oh, well, you know, these parts worked really well for a boat. I think I'm going to make my airplane out of the same stuff. You would never do that, right? You would find parts that are more appropriate for an airplane and build it separately. You would not expect the same design for a boat that you would use for an airplane. So this evidence of homologous structures is really more consistent with the idea of descent from a common ancestor than it is from being independently created by a creator. And we've talked about this Linnaean system of classification where we've got a hierarchical structure of groups of organisms that share more and more similarities as you go down the line. Organisms that are all very, very, very similar to each other are grouped in the same species. Groups of similar species are grouped together in a genus. Groups of genera that are very similar to each other are put together in the same family. And the reason that this works is descent with modification. Descent with modification is responsible for this pattern. So all of these species that are grouped in a genus are similar to each other because they all evolved from the same common ancestor, and they all inherited those similarities from the same common ancestor. All of the genera that are grouped into the same family because they're very similar to each other share a more distant common ancestor, but they share all those features in common with each other because they all inherited it together from the same common ancestor. And the same thing with the homologies that we see in the forelimbs of mammals. This is all due to common ancestry, and so we have the idea of homology. The definition of homology is a trait that's shared by two organisms as a result of inheritance from a common ancestor. And so, as you saw in the film Your Inner Fish, the reason the common ancestor that we're talking about here is something very closely related to this fossil that was found, Tiktaalik rosei, which is the closest known fossil relative to the tetrapods, or all of the land-living vertebrates. And if we look at that forelimb structure, we can see Tiktaalik here in the middle, same pattern, one bone, two bones, many bones that we see in living vertebrates. Again, one bone, two bones, many bones. But also, we can even find the same bone structure, homologous bone structure, in the more distant relatives, the lobefin fish that we see in Tiktaalik and in vertebrates. We can homologize these bones with the humerus, radius and ulna, and carpals, metacarpals, and phalanges. So we see the same pattern repeating in groups of closely related vertebrates. The converse of this also argues very strongly for descent with modification. So we can also look at convergent evolution or convergent structures. And a good example of this is the shark and the whale. So both of them have adaptations to living in the water. They're very streamlined in shape. The whale, as you know, does not have hind limbs, both hairless, but the whale, of course, is a mammal. It has to come up to the surface to breathe air, has to give birth to live young and feed them in the water. This would really be unexpected if it was created by a creator specifically for living in the water. It might make you wonder why a creator would say, okay, sharks, you can just stay in the water and do everything you need to do without ever having the inconvenience of having to come to the surface and breathe and take care of air-breathing babies, whereas the whale, you're going to have to go through all this rigmarole associated with being a mammal to be able to survive in the water. It really is much better explained by its descent from land-living, air-breathing mammals. We also see homology at the molecular level, and this is where things get really, really interesting. But you would not necessarily expect to see similarities in, for example, non-coding DNA, which we do, in fact, find. But I think one of the most striking examples that really helps to show us how all living things are united in this tree of life are things such as homeobox genes, homeotic genes, that are regulatory genes that direct the development of the body axis from the head end to the tail end, anterior to posterior development. And these genes are highly conserved, and we find them both in Drosophila and exactly homologous versions of these genes in mammals such as mice. So these are very, very, very ancient, highly conserved genes with a very important function that unite these groups that are as distantly related as fruit flies, which are protostomes, and mice, which are deuterostomes. And our final contrasting prediction that we see between special creation and descent with modification is this assumption that Earth and life are relatively recent under the special creation model, and the prediction that the Earth and life are going to be very ancient according to descent with modification. That's absolutely required for this slow, gradual process to happen. And so the evidence for this begins with this idea of uniformitarianism, which led to much older estimates of the age of the Earth. Remember, if we're going to attribute the large geological features that we see in the world to these very slow, gradual processes such as erosion, sedimentation, things like that, very long periods of time are required to explain the features that we see in terms of processes that we can observe in our own human lifetimes. We also see in the fossil record discovery of similar fossil forms in different strata of the Earth. You heard about this in the video about radiometric dating. We have relative ages of different strata of the Earth that tell us that life on Earth is really quite old. And finally, in the modern age, the development of radiometric dating techniques can give us absolute ages of rocks and fossils, and we can be very highly confident in our estimates of the age of the Earth at somewhere in the range of 5 billion years old, and the age of life being somewhere around 4 billion years old. So now, between all of the readings you've done, the videos you've watched, you have lots and lots and lots of examples, and this is really still only just scratching the surface of the evidence that we actually have for this pattern of evolution due to descent with modification. And so what I want you to do with this web lecture is use it as a framework for organizing all of this evidence and thinking about it as addressing specific predictions that are made by the proposition of descent with modification. So what I'd like you to be thinking about doing is taking all of this evidence together and putting it together into a cohesive argument, and that is something that we will be working on in class this week.